Our lecture today is about cell division. By the end of the lecture, the student should be able to define the concept and importance of cell division, describe the mechanism of cell division, differentiate between the two types of cell division, describe the different phases of the cell cycle, which is related, of course, with cell division, describe mitosis and its phases, explain the role of mitosis in the embryonic development, describe the factors affecting the rate of cell division, classify the adult somatic cells according to their mitotic activity, describe meiosis and its phases, differentiate between meiosis and in female and male, explain abnormal meiotic division, its causes and the associated congenital malformations, explain the relationship between maternal age and teratogenesis, and explain how the process of hyperplasia, the process of hypoplasia and aplasia. Now we start with cell division. Of course, this is the first. These are the lectures of the reference books which you may need to refer to. The best one, of course, is Ham's Histology. This is the best reference book in the world, and histology, of course. And after that, we start the concept and importance of cell division and the mechanism of cell division. Now, as you can see now, this is a diagrammatic representation of the process of cell division. As you can see here, 2 cells divide to give rise to 4, and 4 cells divide to give rise to 8. Now, let us put this in words. We say that cell division is the multiplication of cells from pre-existing cells. Another diagram here you can see shows something different. The number of cells here are 8, here they are about 7. That means 7 cells. Here you have got 1 cell which has got 2 nuclei. This leads us to deduce that the process of a cell division involves 2 things, the karyokinesis and cytokinesis. By karyokinesis we mean the division of the nucleus, and cytokinesis the division of the cytoplasm. In this case here, what happened? The nucleus divided, but the cytoplasm remained intact. These are the historical figures of histology who contributed a lot in the formulation of the cell theory. Now, this gentleman here, German medical doctor Rudolf Virchow, he worked a lot in cell division, and together they formulated the cell theory, which states that all living cells are made up of cells. Cells are the basic unit of structure and function in organism, and cells come from the reproduction of existing cells. This is, of course, the cell division. Then after that, we come to what we have explained before. Now, in each slide, I have prepared a number of questions. For example, we have already discussed these slides. Let us see what questions have we prepared for it. You can see here questions on this slide. Each slide has about 3 to 4 to 5 to 6 questions. I'm going to prepare all these slides in a special PDF, which will come annexed to the lecture. For example, the first question, define the term cell division. The second question, differentiate between karyokinesis and cytokinesis. The third one, explain the mechanism of the formation of binucleated cell. And the fourth question, explain the mechanism of the formation of multinucleated cells. And number 5, differentiate between proliferation and differentiation. These questions have to be prepared and answered by you, and then you will send them back to me. Now, the second point, or the second topic, the third topic here is how many types of cell division. We have got two types of cell division. One of them is known as mitosis and the other one is known as meiosis. The difference, or the major difference between the two, is that meiosis or mitosis is just one single division, while meiosis are two consecutive cell divisions. Now, these two cells are identically, these cells are not identical. Now, what is the definition of mitosis? Mitosis is defined as one cell division involving equitable distribution of nuclear and cytoplasmic materials, leading to the formation of two identical daughter cells. And these are every cells divide or distributes its nuclear and its cytoplasmic materials. Yeah. And meiosis, of course, is a double division. Here we say meiosis is a special type of cell division made up of two consecutive stages leading to the formation of gametes. This stages is involved in the formation of the oocyte, the egg, and the spermatozoans, and it takes place in the testis and the ovary. We will discuss these two topics in more detail. Then, of course, meiosis, you can see here meiosis, this is meiosis in male. Meiosis in male is involved in the formation of the spermatozoas. Sometimes we call it spermatogenesis, or the process of the formation of spermatozoon. While this one is involved in the formation of the egg or the oocyte. Sometimes this is referred to as oogenesis, and we have got the three polar bodies. We will discuss this in more details, of course, in the later chapters. Here I've got a question, of course. Mention the differences between mitosis and meiosis. One question you already know is that this is one division, mitosis is one division, and the meiosis represents two consecutive divisions. Then we have the so-called cell cycle. The cell cycle, as you can see here, incorporates two major phases. It incorporates two major phases. One phase is known as the interphase, which has sub-phases, which are the G1, S, and G2 phases, and then you have got the cell division or mitosis. As you can see here, this is the G1 phase, the S phase, and the G2 phase. These three represent the interphase. And here you have got the mitotic phase, in which you have got the four stages: prophase, metaphase, anaphase, and telophase. Now, the G1 stage is the stage during which the cell grows and does its functions. The cells, most of your cells are in the stage of G1. This G1, of course, begins after mitosis, and it is the longest and also the most variable. Some of them take hours, some of them take days, some of them are always in the G1 stage. Like, for example, the nerve cell. The nerve cell is always in the G1, always in the G1 stage. Then you have got another stage which is known as the S stage, or the synthesis stage. This involves the duplication of DNA, which takes hours. Tell me, how many hours does it take to duplicate the DNA? And then you have got the G2 stage, which is a transient stage. The G2 stage is a transient stage. It prepares the cell for the process of cell division, or the process of mitosis. The duration and the checkpoint, we're going to discuss this now. Now, coming to the G1 stage, this is the G1 stage. We say that the G1 stage begins, it's the longest and it's variable. Here we form RNA and proteins. The question I'm asking you, synthesis of RNA and protein for what? Second question, mention the cells which are in G0. What do you mean by G0? These things you have got to fetch in your reference books and bring the answers to me. 3. Mention the functions of the two checkpoints. You consider, لاحظ هنا, as we say in Arabic, consider, we have two checkpoints, G1, and we have got another checkpoint which is known as the restriction point. What are the functions of these checkpoints which are found in the stage of G1 stage? Coming to the G2 stage, how long does the cell remain in G2? Does it take minutes? Does it take hours? Does it take days? Find out yourself. Another question, of course, mention the functions of the two checkpoints. Here I've got two checkpoints, of course, in the stage of G2. Then the third thing, or the third point, of course, is the third question involves the mitotic phase. Mention the function of the two checkpoints which are found in the M stage. M stage. You have got number of reference books which are going to be very helpful to you. Try to refer to them and answer all these questions. I have about 50 questions in total, and I'll bring them all to you in PDF. But these questions are only for this slide. Then describe the term mitosis and describe its phases. Here we have the cell mitosis or cell division. Mitosis involves the equitable distribution of cytoplasmic and nuclear material to two daughter cells, which are identical. You have got four phases, of course, in mitosis. The first phase is known as the prophase. What is going on in the prophase? Tell me what changes are taking place in the nucleus and what changes are taking place in the cytoplasm. In the nucleus, you'll find the appearance of the chromosomes. The chromosomes start to appear. Before, during the interphase, we had what is known as the chromatin. Now, this chromatin has been transformed to chromosomes. These chromosomes are, of course, you have got the chromatase and you have got the centromere. In the cytoplasm, you have got the appearance of the mitotic spindle. And the mitotic spindle, of course, you have got the centriole, and here you have got what are known as the microtubules. The second stage is a very important stage, and you can see that in the second stage, all the chromosomes are arranged in the equatorial plane. They are arranged in the equatorial plane, in the center. You can see here that you have got two types of microtubules. You have got microtubules which start from one pole to another pole. You have got other microtubules which connect the chromosome to the pole. Therefore, you have got two types of chromosomes here, microtubules here. Number three, what is going on in the number three, you have got what is known as anaphase. During the anaphase, you have the separation of the chromosomes into chromatids. The chromatids start to separate and they go to the opposing poles. This is stage. Consider, here you have got chromosomes. These are the chromosomes. And here I've got the chromatids. And as you can see here, I've got the chromatids. Number four, you have got the last stage, which is known as the telophase. The telophase is the last phase during which you are going to see the cell is going to separate into two intact units. Now, this is a cleavage. What is the cause of cleavage? I have a number of questions, of course, regarding the mitosis. Here, you have got mitosis, for example. For example, in prophase, I have the following questions. Differentiate between chromatin, chromosome, and chromatid. What is the mitotic spindle? What is the mitotic spindle and what is it made up of? What is the mitotic spindle made up of? And what is MTOC? By MTOC, we mean microtubules organizing center. Answer these questions for me. With regard to the metaphase, I have the following questions. How many types of microtubules are present at the metaphase? Differentiate between them. Differentiate between these two types of microtubules which are found. Then differentiate between the prophase and the telophase chromosome. This is a chromosome and this is a chromosome. Differentiate between them. So in size, in position, in configuration, all of that. And then you have got what is known as the anaphase. Differentiate between the metaphase and anaphase microtubules. You can see here that the microtubules are short and here the microtubules are long. This represents what? This represents what is known as the continuous microtubules, and these are the chromosomal microtubules. What causes the chromatid movement to separate opposite poles? How do they move from here to here? From the center to the pole. How do they move from the center to the pole? Then you have got what is the difference between these two structures? Each somatic cell contains 46 chromatids. Is this statement true or is it false? Then the last stage, of course, cleavage and its relation with myofilaments. These are, of course, the questions which belong to mitosis. After that, we have this structure, as you can see here. What does this structure or this diagram depict? As you can see, what is going on? Some globules going on from one part to another part. Okay, this is depolymerizing, this is polymerizing. What do these two cells represent? What do these two structures represent? These are two structures, of course. What do they represent? And they are related with the process of meiosis. Mitosis and meiosis also. The process of mitosis, we concentrate more on mitosis. They say that mitosis is an essential feature of embryonic development. As you know, at the beginning of your life, you were called the zygote. Zygote is made up of 1 cell. Now, how many cells do you have? Quantitatively, as an adult, you have got trillions of cells. Quantitatively, you have trillions of cells. Qualitatively, you have more than 200 types of cells. And all these cells are organized into 4 basic units of basic tissues or basic groups. These groups are known as the epithelium, the connective tissue, the muscular tissue, and the nervous tissue. We can see, we start here. We start with fertilization. See, this is the zygote. It is dividing into 2, and then into 2, into 4, into 8, into 16, as you can see here, until you form what is known as the morula. You will be studying this more in embryology. Now, this stage all is mitosis, 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 and mitosis. Now, the first thing, this all takes place during the first week of development, during which starts with fertilization. By fertilization, we mean the fusion of the male and female cells or gametes to form a zygote. The zygote duplicates its DNA, undergoes cleavage. This is the cleavage. This is the cleavage. This is the cleavage of this. All is the cleavage. Then the cleavage forms the morula, and then the morula is transformed to blastocyst, and the blastocyst starts to penetrate here in a process known as implantation. Is known as implantation. All of this takes place in the first week of development. Then you have the second week of development, during which you form the bilaminar germ disc. Don't worry about the details of this. I have something in mind. Then you have in the third week the formation of the trilaminar germ disc, which is made up of ectoderm, mesoderm, endoderm. Now we want to know our four basic tissues: epithelium, connective tissue, muscular tissue, and the nervous tissue. From where do they come? The ectoderm will give rise to what? Epithelium, of course. Mesoderm will also give rise to epithelium. Examples are the endothelium and mesothelium. The endoderm gives rise to epithelium. Therefore, we can say that the epithelium arises from the three germ layers: ectoderm, mesoderm, and endoderm. Now, the ectoderm also gives rise to the nervous tissue. The mesoderm will give rise to the muscular tissue, and it will give rise also to the connective tissue. And I have made more than one question mark. Why did I put a question mark here? Find out, because this topic is a bit controversial. What is the real origin of connective tissue? Is it only mesoderm, or is it mesoderm and some other germ layer? Now, these are the origins of the four basic tissues. Factors. What factors affect the rate of mitosis? What are the factors which affect the rate of mitosis? One of these factors is the site. What do you mean by site? The site is the place. The site is the venue. For example, in the brain, do you have mitosis? For example, how in the mitosis of the brain, there are cells of the brain which do not divide. There are cells of the brain which divide. For example, you have got the neuroglia cells, which are the supporting cells, and you have the neurons, and you have the neurons which do not divide. If you go to the small intestine, and if you go to the stomach, you find that the lifespan of most of these cells is around 3 to 4 days, after which they die and then they must be replaced. The second factor which affects the rate of cell division is demand. What do you mean by demand? It means that if the liver, when it is intact, it rarely divides. The cells of the liver rarely divide when they are intact. But when you cut a portion of the liver, you will find that the cell division is very, very rapid. This example of this is the liver. And then you have got chemical factors also affect the cell division. These chemical factors, colchicine, vinblastine, what do? They stop cell division. They stop cell division. For example, you have here, they inhibit the formation of the mitotic spindle or the spindle microtubules. They arrest the cell division at the stage of metaphase. Colchicine. You have got another factor which can stop cell division. This is known as the ionizing radiation. And radiation, of course, in the big doses will stop cell division. And also, it is involved. You can see here, this is the inhibition. This is when they stop the colchicine and vinblastine. When they stop cell division, they stop them at this stage, metaphase. Metaphase is a very important stage. During this stage, you can count the number of chromosomes you have. Therefore, we are going to make use. What is the importance of colchicine? We are going to see just now. Let me finish with radiation. Radiation causes congenital malformations. By congenital malformations, we mean that defects which are found in the baby during birth. Now, we have, for example, I have the first. These are cases of Down syndrome. The Down syndrome is due to increase in one of the chromosomes. For example, in chromosome 21, every cell is supposed to have two chromosomes. This cell has got 3 chromosomes of type 21, and it is called the Down syndrome. Your problem here is a congenital malformations. Now, how do you know? How can we diagnose that this case is present in a pregnant woman? We have to do what is known as the karyotype. Karyotype, and we use the phytohemagglutinin and colchicine. We're going to see that now. This is a karyotype. What is a karyotype? We want to find out whether this baby is normal or abnormal. Whether this baby is normal or abnormal, or this fetus is normal or abnormal. Now, to do that, you have got to take a portion of the amniotic fluid. This is known as the amniocentesis. This is the yellow secretion, or the yellow fluid. This is taken through the syringe and then put in a test tube. This yellow fluid contains cells which belong to this baby. These are the cells which belong to this baby. Now we are going to study these cells. How do we study them? The first thing we do is we put a chemical substance known as phytohemagglutinin. The phytohemagglutinin is going to cause the division of the cell. These cells will start to divide. They will start to divide. They will start to divide, and then you put another substance known as colchicine, which will stop cell division. It stops cell division at which stage? At the stage of metaphase. At the stage of metaphase. And then we start to count the number of chromosomes, whether this baby is suffering from trisomy or is suffering from monosomy. Trisomy means he has 47 chromosomes. Monosomy means he has 45 chromosomes, 1 less chromosomes. Then we classify, of course, somatic cells. We classify them according to the mitotic activity. Their mitotic activity, the adult in the adult organism, of course. Now, we have, for example, here what is known as the classification of the somatic cells in adult organisms according to their mitotic activity. You have got 3 cases: static cases. You have another cases, stable cell populations, and then you have got the renewing cell populations. Now, we start with the first one. Static means stop, or static means not moving.